IB Bio Cell Division Part 2 will focus on the details of mitosis as an asexual cell division that serves to provide growth and repair for eukaryotic organisms. Remember, the essential idea is cell division is essential but must be controlled. Mitosis is an asexual cell division that's important to growth, embryonic development, and tissue repair, and it is seen here as a part of the human life cycle. Following fertilization, a diploid zygote results, and then, with repeated cell division, mitosis, a multicellular adult forms. Most of you are still developing into your adult form through mitosis. Now, meiosis has its role here in the human life cycle. It's a sexual cell division that occurs in sex organs only, and it produces sex cells, the egg and the sperm, known as gametes. And these cells have half the number of chromosomes, 23. This is known as the haploid number. Gametes will then fuse, sperm and egg fuse, that's known as fertilization, and that follows the mating of the male and the female. The fusion of the haploid gametes restores the diploid number of chromosomes at 46. Meiosis and the subsequent fusion of sperm and egg of mating individuals creates genetic variation, genetic variety. More details on meiosis will be provided in Cell Division Movies, Part 3 and 4. Now let's focus on the cell cycle that includes mitosis. A cell spends most of its time in G1, a growth phase, where the DNA is in a diffuse state and the cell undergoes typical cell activities such as protein synthesis, cell respiration, photosynthesis, fermentation, etc. The DNA in G1 is not replicated. Many cells in the body spend most of their time in G1. Now, if a cell is to divide, to undergo mitosis, the DNA replicates during the S phase. Then the cell has a brief second growth phase, G2, that prepares the cell for mitosis. G1, S, G2 together are known as interphase. Technically, mitosis is the division of the nucleus, and that's seen right here. There are 4 parts of mitosis: prophase, metaphase, anaphase, and telophase. And then, after mitosis, the cytoplasm divides, and that's known as cytokinesis. Here are the IB syllabus statements. Outline the stages in the cell cycle, including interphase, G1-SG2, mitosis, and cytokinesis. State that cytokinesis occurs after mitosis. Here's another diagram of the cell cycle. Most cells spend most of their time in the G1 phase, where all of the normal activities of a cell occur. If a cell is about to step into mitosis or meiosis, then the DNA must be replicated during the S phase. Then there's a brief second growth phase prior to cell division. As cell division begins, prophase is followed by metaphase, followed by anaphase, and then telophase. All of this is mitosis. Mitosis is then followed by division of the cytoplasm, known as cytokinesis. Remember that G1S and G2 are interphase. One way of understanding mitosis is to use this graph, which shows the amount of DNA per cell on the y-axis across the time of the cell cycle. At G1, the amount of DNA per cell is this quantity in arbitrary units, and then with S phase, the DNA replicates, it doubles. To have this quantity of DNA, it's double the amount in G1. In G2, the cell prepares the nucleus to divide, spindle fibers form, and then the cell goes into prophase, where the DNA condenses. Then metaphase, anaphase, telophase to result in 2 genetically identical cells, each with the same complement of DNA as the original cell. After the first cycle, the 2 cells are in G1 here, with the potential of another division cycle ahead. Remember that G1S and G2 together are known as interphase. In this micrograph of onion root cells, we can see various stages of cell division. Some cells are in interphase, seen here. Notice one cannot see distinct chromosomes because the DNA is in a diffuse state. It's not supercoiled, it's not condensed. The DNA in interphase is called chromatin. Some cells are in prophase, seen here. Chromosomes can be seen as distinct structures. Some cells are in metaphase and anaphase and telophase, in that order before cytokinesis, which is a division of the cytoplasm that follows mitosis. I've returned to an image of the 2 chromatid chromosome to remind you that chromosomes are replicated during S phase of interphase to become the 2 chromatid chromosome, and they're condensed during prophase to become visible, as we can see here. Chromosomes look like this during prophase and metaphase, but during anaphase, the genetically identical sister chromatids are separated into separate chromosomes, each being moved into separate nuclei of different cells. Here's an IB syllabus statement. State that interphase is an active period in the life of a cell when many metabolic reactions occur, including protein synthesis, DNA replication, and an increase in the number of mitochondria and chloroplasts. Here's a nice image showing the phases of the cell cycle, including interphase and mitosis. Interphase can be seen here, and the phases of mitosis through here. Remember that the DNA replicates during interphase and then condenses in prophase. And then in metaphase, the chromosomes line up in a single straight line, readying to have the sister chromatids pulled apart into separate chromosomes during anaphase, and then ultimately moved into separate nuclei during telophase, followed by cytokinesis, which is a division of the cytoplasm. I hope you notice that this is mitosis in a plant cell. The regular shape and the cell wall should be clear on this point. Here's a micrograph of onion root cells again. Interphase is seen here, here, and here. The DNA is diffuse. It is not supercoiled, thus the chromosomes are not visible as structures. Furthermore, in S phase of interphase, the DNA doubles, the DNA replicates. As we step toward the details of each of the 4 phases in mitosis, here's the syllabus statement that's important. Describe the events that occur in the 4 phases of mitosis, include supercoiling of chromosomes, attachment of spindle microtubules to the centromeres, the splitting of the centromeres, the movement of sister chromosomes to opposite poles, and the breakage and reformation of nuclear membranes. Describe, using diagrams, the process of mitosis, all phases. With this slide, I'm introducing you to prophase, and here are the important events of prophase: the supercoiling of chromosomes, such that they become visible to us within the nucleus of the cell during this phase. Chromosome centromere attaches to spindle microtubules, and there's a breakdown of the nuclear membrane, which can actually be shown here in this drawing of prophase. Remember that chromosomes are composed of DNA and protein, histone protein, around which the DNA is wrapped, forming nucleosomes. And during prophase, the chromatin is condensed, supercoiled into chromosomes. Here's another look at prophase. We can see the X-shaped 2 chromatid chromosomes. The chromatin condenses during prophase. The nuclear envelope breaks down. 2 spindle fibers attach to each chromosome, and the centrioles move to opposite poles. The centrioles play a role in spindle formation. Here is a cell in prophase. The arrows are pointing to visible chromosomes, visible because the DNA wrapped around the histone proteins has supercoiled, condensed during prophase. In this image, I introduce you to metaphase, where the chromosomes line up at the cell equator, at the center of the cell, in a single line, as can be seen here and here and here. Remember that the spindle microtubules already attached to the chromosomes during prophase, but that as the chromosomes line up in metaphase, there are 2 spindle microtubules attached to each chromosome, 1 on each side. We can better see the microtubule attachment in this image. There are 2 microtubules attached per chromosome, 1 microtubule on each side, so that each sister chromatid will be moved to an opposite pole. The lineup of the chromosomes as a single straight line along the cell's equator, along with the microtubules, ensures the formation of genetically identical nuclei. In this image, we can see the DNA wrapped around the histone protein to form nucleosomes, that is slightly coiled to produce chromatin, that becomes supercoiled to form chromosomes, and that the two-chromatid chromosome has two spindle fibers attached to it, one on each side, and this ensures that the two genetically identical sister chromatids are pulled apart, one to each pole, to ensure that the two nuclei formed after mitosis is over are genetically identical. In this image, I introduce anaphase. You can see cells in anaphase here and here. Here's a diagram of a cell in anaphase and an electron micrograph of the chromosomes being pulled to opposite poles. During anaphase, the centromeres break, allowing the chromatids to become full-fledged chromosomes and then moved to opposite poles. The microtubules are involved in moving those chromosomes to opposite poles into separate nuclei. Here's another look at anaphase. The centromere holding the two sister chromatids together as a single chromosome is broken, allowing the now separate chromosomes to be moved toward opposite poles in anaphase. And here we have telophase. In telophase, the nuclear membranes begin to reform around each of the two new nuclei. As well, the chromosomes begin to decondense, becoming chromatin. In plant cells, the cell wall begins to form, as you can see here, while in animal cells, the membrane pinches in, as you can see here. Telophase is the final stage before cytokinesis. Here's an image of telophase in an animal cell. You can see the nuclear membrane beginning to reform here and here, as well as the beginnings of a nucleolus in each nucleus. And the membrane is pinching in prior to cytokinesis. This pinching in of the membrane is known as a cleavage furrow. Here are the IB syllabus statements of relevance. Define the term mitosis. You need to refer to a division of the nucleus here. State that cytokinesis occurs after mitosis. That's relatively straightforward. Cytokinesis occurs after telophase of mitosis. And distinguish between cytokinesis in animal and plant cells. Realize that animal cells have a cleavage furrow as the membrane pinches in, and plant cells form new cell wall along what's called the cell plate. In this image of cytokinesis in a plant cell, we can see two new nuclei here with the separation of cytoplasm occurring with the cytokinesis process. The cell plate is the formation of new plant cell wall, which will be extracellular cellulose. In this image, we can see cytokinesis in animal cells. The cleavage furrow is very clear as the cell membrane pinches in the cytokinesis process. Now, as we bring our close examination of mitosis to a close, I want to remind you of the essential idea, which is cell division is essential but must be controlled. Cell division is a fundamental reproductive process, and the ability to reproduce, to have cells arise from pre-existing cells, defines living things like no other characteristic would define living things. However, if cell division is not controlled, tumors result, cancer. Let's take a look at some of the detail around the control of cell division. Here are the two relevant IB syllabus statements. Describe tumor formation, which is known as oncogenesis. Refer to oncogenes, mutagens, and metastasis in the formation of primary and secondary tumors. Define the terms oncogenes, mutagens, and metastasis. Mutagens are physical or chemical agents that cause mutations in the DNA. The DNA mutations caused by the mutagens initiate uncontrolled cell division, leading to cancer, tumors. Mutagens include X-rays, UV light, and various chemicals such as cigarette smoke or even exposure to certain bacteria and viruses. During cell division, DNA replicates. You can see the replication process here. During replication, mutations can occur because the DNA is exposed as it is being copied by an enzyme. The mutation is a copying mistake with the insertion of an improper base. The base pair on this newly forming strand should be identical to the base pair on this newly forming strand. The mutation has resulted in a genetic difference. Remember, mutagens increase the rate of mutation. If the mutation occurs in a gene that normally controls cell division, then uncontrolled cell division, cancer, may result. Oncogenes are genes that control cell division. In other words, oncogenes normally inhibit uncontrolled cell division. Oncogenes inhibit tumor formation. But if oncogenes become mutated, as can be seen on the right here, due to the exposure to mutagens, then cancer, uncontrolled cell division, results. As you now know, uncontrolled cell division results in the development of a tumor, a lump of cells with characteristics of the original tissue, but without the normal control of cell division. A primary tumor is found in the original tissue in which the tumor started. But if a cancer cell escapes the original tissue, as can be seen here, and enters the bloodstream, as can be seen here, and travels to a distant location in the body and then grows, metastasis has occurred. Metastasis is defined as the development of secondary growths at a distance from the primary site of cancer. The secondary tumor then grows and spreads rapidly, and the diagnosis of malignancy is not good news for a patient. Here are some definitions that I've already defined earlier in this movie. I will let you study this slide on your own. And here is one last IB syllabus statement related to the control of cell division. Describe briefly how cyclins control the cell cycle. Cyclins are proteins. As proteins, the synthesis of which is directed by DNA, if that DNA that directs cyclin synthesis becomes mutated, cell division may no longer be controlled. Now this statement is one you've already seen. Describe tumor formation, oncogenesis, refer to oncogenes, mutagens, and metastasis in the formation of primary and secondary tumors. Now cyclins are proteins present in high concentrations in the cytoplasm during cell division. You can see the concentration of different cyclin proteins rising and falling, rising and falling, rising and falling through the stages of mitosis. The cyclic nature of the concentration change is the origin of the name cyclin. Now the inference from the changing concentration of these proteins, the cyclins, is that cyclins play a role in regulating cell division. Cyclins are not enzymes, but cyclins do influence enzymes important in cell division. For example, cyclins influence the initiation of DNA replication and spindle formation. Now here's some information about tumor formation. Tumor formation, oncogenesis, results from mutations in the genes that control the cell cycle. The genes are called oncogenes. And anything that increases the risk of mutation increases the risk of tumor formation, and these would be the mutagens such as chemicals or radiation. Mutagens could be chemicals in cigarette smoke as well. When cell cycle control is lost, the cell undergoes repeated cell divisions that produce a mass of cells known as a tumor. This is a primary tumor, and usually it's benign. In other words, it has yet to go through metastasis. The spreading of cancerous cells in the body is known as metastasis. The tumors that develop from the spread of cancerous cells are known as secondary tumors. If secondary tumors are not treated, they can lead to death. In this diagram on the left, we see the lungs of a smoker with cancer, seen as the white lumps here and here. In the photograph on the right, we can see cancer here. Here is a photograph that compares the lungs of a non-smoker with a smoker. Here is a graph that displays a positive correlation between smoking and lung cancer. As with all correlations, the interpretation must be handled carefully because correlation does not necessarily mean causation. For example, just because cigarettes have been purchased doesn't necessarily mean that they've been smoked such that they would lead to cancer. However, many studies support the correlation between smoking and lung cancer, as you can see here. And while we must always be careful not to assume correlation equals causation, many studies have implicated specific chemicals in cigarette smoke as mutagens. Thus, scientists have confidence that the correlation seen here represents causation. This is a graph that displays survivorship over lifespan, and the information suggests that those who smoke, seen in these blue lines, have a shorter lifespan. The glossy presentation of this graph makes me suspicious of political bias in reporting. It's not that I think smoking is healthful, but people of science must always be skeptical, and especially when data appears to have a political motive, such as this one. Now, the mitotic index is a tool of oncologists, cancer specialists, who would diagnose cancer in someone who is suffering. The mitotic index is a simple count of cells in the stages of mitosis divided by the total number of cells counted. If the percentage is high in the sample tissue, cancer would be suspected. As well, the index can be used to assess the effect of chemotherapy. If chemotherapy was effective, then the mitotic index would be lower than it had been prior to the therapy. Here's another image displaying the calculation of the mitotic index. Study this. It's something that you need to know. Can you identify the mitotic stages shown in the image? 3 is prophase, 1 is metaphase, 2 is anaphase, and 4 is telophase. Thus, as we bring this movie to an end, can you explain how mitosis produces 2 genetically identical nuclei? Start with DNA replication that happens during the S phase of interphase, the formation of 2 genetically identical pieces of DNA that are attached at the centromere, then 2 spindle microtubules, one on each side of the 2-chromatid chromosome, move the chromosomes to the metaphase plate to form a single straight line of chromosomes. Then the spindle microtubules pull the genetically identical sister chromatids apart so that each becomes a separate chromosome, moving to opposite sides of the cell into 2 different nuclei formed in telophase and then separated in cytokinesis. Now remember, mitosis is an asexual eukaryotic cell division process resulting in genetically identical cells. Mitosis is important in growth, embryonic development, and tissue repair. Meiosis, on the other hand, is part of a sexual reproduction process involving the union of gametes, sperm, and egg from different individuals. Meiosis increases genetic variation in a population, while mitosis does not. And that brings us to the end of IB Bio Cell Division, Part 2, Mitosis, Part 2.